welcome to this instructional video for Dr. Jabbour's lab. Today we'll be doing a short video about the basic protocol for immunohistochemistry in the lab. Let's start. So the first thing I did was I went and got some slides from the blue boxes. Today I'm going to be running basal nuclei. So these three are each controls, and then case 93 is a test case. That's an opioid overdose case. So the first thing we're going to do is we're going to start dewaxing. Here we have a jar of Citrazolve. These were actually just changed, so make sure you change these every 5 times you use them. So it's easiest to use your hands. You can also use a pair of forceps if you want. And all we're going to do is we're just going to put these in. And you want to line them up. There's ridges on the sides. You're going to want to line them up with those. And try your best to make sure that the slides don't touch. Alright, so now that those are in, I'm going to set a timer for 2 minutes, and we'll wait. So the timer just went off, and now we're going to put it into our second bath. I'm going to use forceps this time. So the important thing here is you're going to want to pull out without the slide touching the other slide. So you're going to want to pull out straight and just put it in the second jar, just like that. Again, making sure that the ridges align with the slide edges, and making sure that the slides don't touch once they're in the jar. So for time's sake, I'm not going to show you the change of every single wash for dewaxing. It's the same thing. You'll notice that the first two are Citrazolve, and then it goes from 100% ethanol all the way down. It's decreasing percentages of ethanol, and then until you get to pure water, and then you get down to PBS. Once you're in PBS, you're going to come over here to the pressure cooker, and you're going to turn on the pressure cooker. Alright, so I just put the slides into PBS. I've done all of these dewaxing jars. I have about a minute left on this one. So I'm going to come over here, and I'm going to plug in the pressure cooker. So here's the cable. I'm just going to plug it in. And then what you want to do is you want to click pressure adjust, and it's going to start heating it up. Alright, so steam is just now starting to rise out of the pressure cooker, and if you can see there's some bubbles around the edge. Make sure you check when you're doing this that the water level is high enough that it won't boil away and burn all the slides. I've done that before. Alright, so what I'm now going to do is I'm going to take the slides out of the jar one by one, and you're going to arrange them in a sort of square shape face up so that they're not touching and that they're actually as far away from each other as you can get them. Alright, so the slides are in. They're as far away from each other as they can be. They're all face up, and now I'm going to put on the lid. So you just put it on like that, and then you lock it, and then you're going to want to close the top. And now we're just going to wait 10 minutes while the pressure cooker does its thing. While the pressure cooker is going, as you can see we have 7 minutes left on it. We're going to start thawing our antibody. So what you're going to do is you're going to come over here. There's all these containers with cells in them, so we're going to grab one of those. You're going to come over here. I already made ice and put it in here. You can grab that in the prep room if you want. And you're just going to put that in there, and now we're going to go to the refrigerator. So there's 2 refrigerators. There's the 4 degrees C and the minus 20 degrees C. So our antibody is going to be in the minus 20. So we're going to go down here, and it's this block from cell signaling. So we're going to take that out. So we're going to open up this box, and here's our antibody. And I'm just going to put it in one of these. So the reason that we cool it on ice is so that it doesn't thaw too quickly and damage the antibody. Alright, the pressure cooker just beeps to let me know it's done. So what I'm going to do is I'm going to take a pair of forceps, you can also use a pen or something, and you're going to come up to the button, you're going to slide it forward, and it'll release the steam. Just for reference, this is the button that you're going to slide forward. It was pushed down, then you're going to push it a little forward. Alright, so now we're going to take off the lid, unlock, and lift off, and then we're going to put it down. And inside, as you can see, every slide is fine. They're all face up, and they're okay, so we're going to start taking them out and putting them back into PBS. Alright, we have another jar of PBS here. That also needs to be changed every 5 times you use it. And it's cool, room temperature. So we're going to start cooling off these slides by taking them out carefully. They're very hot, so you're going to want to do this with forceps as well. And you're going to want to put these in the PBS. Again, like the dewaxing jars, you're going to make sure you line them up with the ridges and making sure the slides don't touch. So all the slides are in the jar now, so we're going to cap it, set a timer for 30 minutes, and go listen to some music or something. While those are cooling, we're going to come back to our antibody, and I like to gently rock it to see if it's thawed or not. And this one looks pretty thawed because it's moving while I move it. So I'm going to put it back on ice, and we're going to spin it down. So to spin down the antibody, we're going to come over here to this drawer. We're going to get an aliquot. We're going to cap it. This is going to be our counterweight so that the centrifuge doesn't spin itself and explode. And we're going to grab our antibody. We're going to come over to the other side of the lab and put it in the centrifuge. So you always want to make sure that the centrifuge is lined up. So numbers 1 and 10 are lined up. Same with 2 and 11, 3 and 12, etc. And then you're going to put the cap on until it clicks. And then you're going to bring the door down. It's set for half a minute or 30 seconds. And you're going to let it go. So the centrifuge just stopped spinning. I'm going to take the cap off. I'm going to take the antibody out. And I'm going to leave the counterweight in for right now so that I can walk the antibody all the way over and put it back on ice and let it chill for a little bit. So next you're going to go to the fridge and you're going to get antibody incubation solution. The recipe for this is on the master list. So you're going to open that up. And you're going to carefully pipette 798 microliters into each of 4 aliquots, which I have here. Alright, and you can use the same blue tip for that. And then you're going to dispose of the tip, close your box, cap your solution, and return it to the fridge. Next you're going to get the 2 to 20 microliter pipette, and you're going to get a box of yellow tips. This is actually the hardest part of... Okay, take 2. Next you're going to get a 2 to 20 microliter micropipette and a box of yellow tips to match the yellow top to your pipette. And what you're going to do is you're going to uncap the antibody, which has now been spun. It's the most important part. You're going to open your box of tips, and you're going to draw out 2 microliters of antibody. You should be able to see it in the tip there. I don't know if it'll pick it up on the footage, but you should be able to see it. And you're gonna put in 2 microliters, and you have to make sure you change the tip every time. Alright, so now that that's done, you're going to take your antibody, cap it, and return it to the ice. Alright, so there's no point in leaving the antibody out now, so we're just going to put it back in the box, just slide it out, put it in one of the middle ones, back in the box, and return this to the fridge. Alright, these slides just finished cooling down to room temperature. It's been 30 minutes, so what you're going to do is you're going to grab the Hydropen from the fridge, and you're going to grab some Kim wipes, and we're going to start IHC. So what you're going to do is you're going to take them out. I like using a pair of forceps and then transferring to my fingers. So you're going to take it out. Now it's in your hands, and you're going to dry off the bottom of the slide. Make sure you don't touch the top, because obviously that's where the cells are. And you're just going to want to wipe off the PBS from the corners and the edges, but it's really important that you don't touch the middle and that you don't dry out the middle either, because that's where the cells are and you want to keep those wet. Alright, so now what we're going to do is we're going to uncap the Hydropen, and you're going to want to create a border around the slide. Alright, so that's done. And what we're going to be doing is, you can see, probably doesn't pick up well, but there's a hydrophobic border. This is an oil pen, and what it does is it makes it so that all of our solutions don't roll off of the slides. And next we're going to put it onto this tray, which is just a dyeing tray with the canopy turned upside down. Alright, so those slides are all now dried and hydropenned, and there's a hydrophobic barrier around each one. And now what we're going to do is we're going to apply 1,000 microliters, there it is. I'm going to apply 1,000 microliters of this peroxidase suppressor to it. And now we're going to turn on the rocker. Alright, so it's been 10 minutes. These have been rocking and there's been no spillage, so I'm going to turn this off. And we're going to dump these out into the sink. And then what I like to do is take a Kim wipe and just make sure that there's no liquid on the bottom. And then I return them. Alright, so these have all been dumped off and the bottom's been dried, and now what we're going to do is we're going to apply our wash solution, which is right here. And there's a recipe for that, which is also on the master list. And this isn't any particular amount. You can just basically do any amount that you want as long as it covers the whole slide. And you're going to want to reduce bubbles as much as you can. It's been 5 minutes, and this has been rocking, and there have been no spills, so I'm going to turn it off. And we're going to do the same thing. Dump out into the sink, and wipe the bottom. So they're all poured off and dried at the bottom, so now we can move on to our blocking solution. So it's the same process as the one before. It's 1,000 microliters. We're using the blue pipette tips. And so you're going to draw up 1,000 microliters and apply them to each. Alright, so now we're going to set a timer for 20 minutes and let these rock. It's been 20 minutes now, so we're going to stop the rocker and we're going to pour off the blocking solution. Alright, so now that's done, and we're going to apply another wash. So again, this one doesn't have an amount to it, just enough to cover the slide. And we're going to set that for 5 minutes on the rocker. Okay, so it's been 5 minutes, so we're going to shut off the rocker. And we're going to transfer the slides to this container. Alright, so now we're going to take these slides into 113. Alright, so we're now in room 113. Right here is the refrigerator rocker that we're going to be using. There's several ways you can do this. I have my slides right here. I like to put them in the fridge first and then pipette into the fridge. Some people like to do them on a counter and then carry it over. It's up to you. I'm going to do it in the fridge. Alright, so we're going to start with the refrigerator door open. The slides are on the rocker, and what I'm going to do is I have my pipetter. It's set to 800 microliters, and then we're going to take the aliquots. I'm going to take one aliquot out, and one blue tip, and we're going to apply them left to right or right to left, just as long as you remember which ones you've done. Alright, so now all of my primary antibody is applied. It's in the fridge, the rocker's turned off, so what I'm going to do is I'm going to grab a piece of cling wrap. It's in the drawer in 112. I'm going to put it over the top. I'm going to rubber band to keep it on. And now we're going to leave it overnight. We'll come back tomorrow, I'll finish it up. Alright, so it's day 2 now. I've let these go overnight, and we're going to take them out. So I'm going to hit stop. So a total of 19 hours and 57 minutes. Turn it off, and I'm going to take it off the rocker. Alright, we're back in the lab, and I'm going to take off this plastic wrap. So first I'm going to take off the rubber band. And inside you can see there's just a little bit of spillage, not a huge issue. So we're going to take these off, pour them in the sink, and then put them back on the rocker. So now what we're going to do is we're going to put some IHC wash solution on each of them and wash them for 5 minutes. Alright, so it's been 5 minutes, and now we're just going to pour these off, dry them, and apply our secondary antibody. So our secondary antibody comes from this purple Immpress bottle, and we store it in this conical that's labeled secondary antibody. You don't need a ton of it, you just need 1 milliliter per slide, so that should be enough. Next you're going to take our micropipetter set to 1000 microliters or 1 milliliter, and you're going to use a blue pipette tip, and you're going to apply 1000 microliters to each slide, and we're going to rock that for 45 minutes. Alright, it's been 45 minutes, so we're going to turn off the rocker. We're going to empty the slides into the sink, and we're going to do one more wash with IHC solution. So while that wash is running, we're going to take 4 aliquots. And we're going to add 500 microliters of this brown dab and 500 microliters of this clear dab to each aliquot and mix them up. Alright, so it's been 5 minutes, so we're going to turn off the rocker and we're going to pour these into the sink. And then this is an important step, we're going to rinse them with the pipette over the sink. Now we're going to dry with a Kim wipe, and we're going to put them into a dab-only tray. Alright, now it's time to apply the dab, so I'm going to put on some gloves, because dab is a carcinogen. So my gloves are on, And this is an important step because if you apply the dab directly to the slides, it'll clump up, and then that causes some really brown staining on the slides. So what we've discovered is that if you start the rocker and then apply it, it prevents clumping. So we're going to do that now. Alright, so those are applied. We set them for a total of 3 minutes, so I'm going to set a timer for 2 minutes and 30 seconds because they've already been going for a little bit. Alright, so the bell just rang for the dab, so we're going to shut off our rocker. And we're going to quickly, as quickly as we can, remove these and pour them straight into a conical, collect them, and it's going to be labeled dab. And then you're going to use a squirt bottle to squirt off any excess dab that's on there. Alright, we're on the other side of the sink now, and all of our tissue samples are here. You can see that they're light brown, which is a good sign. That means that our antibody did its job and the dab did its job. So now we're going to counter stain with hematoxylin, and this is going to stain any cells that haven't been stained by the antibodies. So it's purple. I'm just going to apply a little bit over the top. And it only needs to be there for about 10 seconds. And lastly, to speed up the drying process, because these are all covered in water, we're just going to dip them in alcohol, which will dehydrate all the water. And then that'll evaporate into the air.